Welcome to The Explainer. I hope you're ready to focus today, because we're looking at something absolutely incredible. We're talking about the microscopic biological engine that literally powers every single thought you have and every single movement you make. It kind of feels like magic, but I promise you it's just pure, fascinating biology.
Just think about that for a second. You decide to move your hand, and bam, it happens instantly. Have you ever wondered how a signal travels from your brain all the way down to your fingertips in just a fraction of a millisecond? Well, to solve this micro mystery, you might want to grab a pen and paper. We're going to zoom incredibly close into the plasma membrane to decode neural transmission and figure out exactly how ions move to make these nerve impulses happen.
Here's our roadmap for today. We're starting with the speed of thought and the basics of cellular transport, then checking out the cellular environment, diving deep into the sodium-potassium pump itself, and finally, I'll give you a magic exam key to help you absolutely crush your physiology tests. Let's get into it.
Okay, starting off with part 1, downhill versus uphill transport, the battle of cellular energy.
To really understand this, we need to compare two fundamentally different ways molecules move inside our bodies. On one side, you have passive transport. Think of this as a downhill process. Molecules just naturally flow from an area of high concentration to an area of low concentration. It's totally free, it doesn't take any energy, it's literally just like a ball rolling down a hill. But the engine we're looking at today operates in completely the opposite way. It relies on active transport. This is an uphill battle, pushing ions directly against their natural flow. And because it's actively fighting nature, it absolutely has to burn energy to pull this off, specifically using ATP.
Moving on to part 2, the cellular environment, setting the stage for the nerve cell.
To see this active uphill transport doing its thing, we have to set the stage. We divide the environment around the cell into two distinct areas, and you'll definitely need to know these. First, there's the ICF, the intracellular fluid. That's the fluid inside the cell. And then we have the ECF, the extracellular fluid, which is the fluid sitting on the outside. Grasping this boundary is absolutely key to understanding how our nerves communicate.
Now here's where it gets really interesting. When a nerve cell is just resting, it's not actually balanced at all. In this resting state, there's a ton of sodium, or Na+, packed on the outside in the ECF. And conversely, there's a huge concentration of potassium, K+, inside the cell in the ICF. It's a very deliberate, highly maintained imbalance. And the cell desperately needs this tension, so it's always coiled up and ready to fire a signal at a moment's notice.
This perfectly illustrates why fighting nature is so crucial here. If these ions were just left alone to balance out naturally through that free, passive downhill transport, everything would just even out. And if that happened, your nerves would literally go dead. You'd lose all ability to send signals. Your brain couldn't even tell your hand to move.
Which brings us to part 3, the Sodium-Potassium Pump, the biological gatekeeper.
Enter the absolute hero of our story. This pump is an integral membrane protein that acts as a highly regulated gatekeeper to stop our nerves from dying out. Let's break down exactly how it works. First, it burns some ATP energy to grab exactly 3 positive sodium ions and physically pumps them out of the cell into the ECF. At the exact same time, it pulls 2 positive potassium ions from the outside and brings them back inside to the ICF. Now, think about the math there. It's constantly pushing 3 positive charges out, but only bringing 2 positive charges in, so it naturally maintains a net negative charge inside the cell.
And that uneven exchange brings us to a massive physiological concept, the resting membrane potential. This is precisely that negative internal baseline established by this lopsided 3-out, 2-in pumping action. Without this pump working around the clock to keep things slightly negative inside, our entire nervous system would just shut down.
So the implications of this are huge. This constant energy-burning pump isn't just trivia. It's the absolute foundation for your nerve impulses. It's the reason your muscles can contract, and it's essential for maintaining overall homeostasis in your body. It's literally keeping you charged up and ready for action.
Plus, if you don't understand this resting state, you're going to have a really tough time later on. Grasping the resting membrane potential is step 1 before you can tackle the complex phases of an action potential. Things like depolarization, repolarization, and the refractory period, none of those make sense unless you understand the baseline that this pump painstakingly builds.
So let's look at part 4, the magic exam key. Time to nail that physiology paper.
If you want to burn this sequence into your memory forever, here's what you do. First, picture the number 3. This is for our outward-bound sodium ions.
Next, lock in the number 2. This stands for the potassium ions we're dragging back inside.
And then, remember their strict directions. Out and in. It's a very precise exchange.
Put it all together and you get 3, 2, out, in. 3 sodium out, 2 potassium in. Honestly, if you can just remember 3, 2, out, in, you have the core mechanism of active transport in the nerve cell completely locked down.
And guys, here is the golden rule of physiology. If an exam ever asks you to explain the mechanism of a nerve impulse, always start your answer by explaining this exact pump and how it creates the resting potential. It is the rock-solid foundation for everything that happens next in neurotransmission. Start there, and you're golden.
All right, to wrap up today's explainer, I've got a quick challenge for you. Based on everything we just covered about how this pump actively moves ions against the gradient using ATP, is the sodium-potassium pump an example of a carrier protein or a channel protein? Think about how it physically acts as a gatekeeper. Drop your answers in the comments below and keep learning.